Very good evening all. So happy to meet you all back in this awesome session. Yes, everybody. So I hope that all this your results are being out, and I know that you are all in very good preparation for the next upcoming examination. Yes, obviously DBT it is going to be your next target. Yes. Okay, so that is what we are going to see today. We will see in detail about the DBT 2021 question paper. Yes, everybody. So you are all with Josh. Shall we just make it out? Yeah, such that it will be easy for everyone. Yeah. Okay, fantastic everyone. So let's seriously make a very important deal with this. So we are going to see in detail about your DBT questions. Okay, so a little bit about me, maybe many may be knowing me. So I have completed my Ph.D. in Biomaterials after cracking CSIR NET as well as the CENT examination. Have guided so many aspirants, yes, for CSIR as well as the other examination. I do have 15 papers for your credit, and you can follow me at Unacademy for more informations. Alright, and there is going to be a price hike at Unacademy. Okay, so this price hike, yeah, it is going to be a little bit more this time. So this 12-month fees is going to go up from 14,500 to 15,950. Okay, so please don't miss it out. If at all you have not yet subscribed, yes, it's high time that you have to come with the subscription, right? Okay, so this is the current course that is being launched. It is the DBT Jumbo Practice course. Okay, so with this Jumbo Practice course, you will be able to practice all the 10 years of PYQs. Okay, so that is going to seriously make a very good impact on the preparation. Alright, yes. So if at all you have not yet enrolled, please make sure that you are going to enroll for this course. Okay, so let's take out the very first question. A mutation that is generating a termination codon. Very first question of the day. Everybody is ready for this? Yes or no? Come on guys. Yes, everybody. Cheer up for the first question of 2020-21. Yes, you can see major questions are repeated. Yes? Okay. So a mutation that generates a termination codon. It is known as what? Whether it is going to be a missense mutation, yes, or else it is going to serve as a nonsense mutation, sorry, or it is going to be a reverse mutation, or else it is going to serve as a silent mutation. Yes, a mutation that is going to generate a termination codon. Termination codon means what? It is going to end something, isn't it? Yeah, so it is going to end something. That is what is termination. So if I terminate something in between, what it is going to lead to? Yes, that is the question. Whether it is going to be a missense mutation, whether it is going to be a nonsense mutation, a reverse or silent. Everything has a meaning, isn't it? So which could be the correct answer? Yes, everyone. Come on, come on. So the correct answer, oh yes everyone, I couldn't see the chat, just now chat open for me. Yes Ram Prabha, yes correct, it is a nonsense mutation. Yes Anjali, absolutely correct. It is going to be none other than the nonsense mutation. And what happens in case of your missense mutation? Sorry guys, my pen is not working. So what happens in case of your missense mutation? It is going to change, isn't it? So if it is going to change, then it becomes like a missense mutation, isn't it? Yes, if it changes, it becomes a missense, and nonsense means truncated, isn't it? We usually call them as the truncated ones, right? Okay, so it is cut shorter. So that is what is the question also. The mutation which is going to generate the termination codon. Termination codon means it is truncated. That is the correct answer. The next one, it is going to be the reverse mutation. It is a changeover. So that is not the correct answer. Silent mutation, even though the codon is changing, the amino acid or the effect it is not going to change. That is what is called as silence mutation. Do you remember the other name for the silent mutation? Yes, in molecular clock you could have studied that. It is termed as synonymous mutation. Yes, so it is termed as synonymous mutation. Got it? Yes, everybody? It is clear now. Shall we make a move towards the next question? See, I couldn't believe my eyes. This is a 2021 question paper. They have asked such a simple question. So, what is the big lesson you have to learn from that of DBT? You have to act fast. That's all. Yes? All are simple questions. Only thing you should have landed up with the correct question. That is the only point that is needed here. Yes? Which of the following it is going to be the achiral amino acid? Fantastic. Who is achiral here? Alanine? No, alanine is chiral. Glycine. Yes, absolutely. Glycine is achiral. So immediately you should keep revising. What is the nature of alanine? Alanine is non-polar. Yes, glycine is also non-polar. Then phenylalanine. Phenylalanine is aromatic. Fantastic. Next it is going to be the proline. So proline is also non-polar and aliphatic. Okay? So all the things should come to your mind. Okay. So the next is a very interesting and important question from your equation. Yes, we have to read this carefully. Everybody can do this question. Okay, right. So, in an enzymatic reaction, okay, so in an enzymatic reaction, the following equation kinetics, yes, doubling the substrate. So, yes, it is going to get doubled. So, from S, it is going to become 2S. So, whenever S is increased, what happens to V0? V0 will also increase, isn't it? So the rate of the reaction is increased by 2%. When you double the substrate concentration, yes Anjali, fantastic. So this is going to double, no, by 2%. So they are asking which of the following statement it is going to be true. Yes, which of the following statement it is going to be true is the question. Number 1, Km is greater than 2S. Okay, which formula you will use to check this? My God, my pen is not working today. Okay, right. Yes, Anjali, you are right. Yes, but how to derive at this? Yeah, okay. First, you have to think of your equation. What is your equation telling? Yes, V0 is equal to Vmax into S divided by Km plus S. This everybody agrees. V is equal to Vmax into S divided by Km plus S. No doubts in this formula, right? It is the equation. Now what happens based on your question? This S is becoming 2S. Okay. So, my God. Sorry guys, my pen is not working. So, just give me a moment. I'll just try it out like, why the pen is not writing, I don't know. Alright. Never mind. Okay. Oh no, it's not writing. That's okay. We'll cope up with this only. Right. Okay. Alright, just a minute. This alone I'll write and then again we'll come back to the slide only. Yes? Okay, fine. Alright, here it is working. Okay, good. Right. So we all know this equation. V0 is equal to Vmax into S divided by Km plus S. Correct? No? Yes? Okay. So now what has happened? Yes, it is becoming 2S. Okay. Alright. And what happened? The Vmax is also increasing. S has become 2S. Right. So what will happen to this fellow? So this will become S by 2, correct? Even then, this value is still more increased, isn't it? So when this will be still more increased? Can you just guess it? So whenever, yes, so I'm sorry. So whenever your Km, it is going to be very much lesser than that of S by 2. Yes, your S by 2, it is going to be still more higher and higher. Okay, yes, so this S by 2, if it is going to be still more higher and higher, yes, then only still your Vmax will show an increase, or else the increase is not at all possible. So that is why, yes, this condition alone, it is going to work out for this particular question. So your Km should be less than SO2. It is not directly proportional, okay, so you cannot say that it is very much greater, isn't it? This first one you can eliminate immediately. Your Km is greater than S0, what will happen? Your Vmax will decrease, isn't it? This is greater means immediately it will cut off, no, so this will decrease. So decretion is not happening, it is still increasing only, isn't it? Here you see there is the increase in the reaction rate by 2%. So first option you will eliminate immediately. Yes? So likewise, what about the second option? That will also be eliminated immediately. Maybe you could have had a doubt between C and D. Yes, but D should be exactly the correct answer. D should be the correct answer. C cannot be true. Why? Because it says that S by 2, it is lesser than Km. This cannot happen, isn't it? So the correct option is only answer D. Got it? Yeah? Okay, fantastic everyone. Yes, so let us make up a move towards the next important question. So in the next question, we are going to see about for sequencing by your for sequencing of the DNA by the Sanger's method. Okay, by means of the Sanger's method, the chain elongation, it is going to get terminated. Yes, Ash. It is going to get terminated by which of the following? 4-dideoxynucleotide, 3-dideoxynucleotide, 1' 4' dideoxynucleotide, 2' 3' dideoxynucleotide, yes, 2' 4' dideoxynucleotide, 4' 3' dideoxynucleotide, and the options are hidden. Yes, Anjali, Ramprabha, yes, Ansh, correct. So it should be 2' and 3' dideoxy. What is this normal 2' deoxynucleotide? 2' deoxynucleotide, it is called as what is that? 2' deoxynucleotide. Nothing other than your RNA, isn't it? But if one more OH is gone, then it becomes your dideoxy, isn't it? So in case of your DNA, yes, you will have a deoxynucleotide. If it is an RNA, it will have everything, correct? In case of DNA, your 2' OH is lost, and in case of your dideoxynucleotide, your 2' as well as your 3' is lost. Understood? Yes, everyone? Yeah? Okay. So shall we just make a move towards the next question? So the next question is also quite simple. Anybody can answer this question. It is absolutely a simpler version of your question. Okay. So they have given the components in the list 1, yes, and the properties in the list 2. You are going to mix and match. Yes? Okay. Very good, everyone. Come on. Leucine, lysine, glutamic acid, glutamine. Fantastic. Yes, everyone. Yes, Ramprabha, Anjali. Come on. Leucine. Yes, Archana. See, if you take leucine. Leucine is what charge, ma? Leucine. Gap will met is non-polar. Isn't it? Yes. So, leucine is non-polar. A should be 2. What is lysine? Lysine is positively charged. So, B should be 4. By that only I will know that B is the answer. Yes, Anchan Anjali? B is the answer. Leucine is non-polar. Lysine is positively charged. Glutamic acid is negatively charged. Yes, yes, Ramprabha, correct. And then glutamine. Glutamine is uncharged but polar. Like your, yes Anjali. So like your cysteine, 309, glutamine, asparagine, TCS, gynamine. They are all uncharged but they are going to be polar. Got it everyone? Is it clear now? Shall we make a move towards the next question? Okay. So next question is very important. Yes, I can tell you that such questions are quite common from your enzymes. Yes, and also it is a very important question from your biochemistry. Let us see a simple question only. Isomerization. Isomers you could have studied. No, isomerization. Redox reaction. Group transfer. Hydrolysis. Yes, yes, Archana, Ramprabha, and yes, come on. Got it. Too good. Isomerization is rearrangement. Yes. Transfer, yes, Anjali. Good. Yeah. So it's obviously going to be your option B. Everyone got it right. Fantastic. You see the DBT question. DBT questions will not be very complex, but you have to solve it fast. You will not have much time. Okay, so you have to solve it very fast, isn't it? So that is the only challenge you have with the DBT questions. Okay, so the questions will be simple, but only thing you have to solve it fast. Okay, so which one of the following cannot be used for differential gene expression analysis? For differential gene expression analysis, you cannot use them. EST, expression sequence tag data analysis, microarray data analysis, mRNA sequencing data analysis, whole genome sequencing data analysis. Fantastic. Why D? I usually used to tell a story. Yes. So there were 2 ponds. Okay. In one pond, 5 frogs were alive. Okay. In the next pond, 4 frogs were alive and 1 frog was dead. Okay. If simply I am asking you to count the number of frogs, yes, you will count both are equal. That means we are not counting the live ones. So this is the total presence. They may be present, but out of them how many is live? That is what is gene expression. Gene may be present. Is it expressing or not? Same thing. If you have a whole genome sequencing data, it will indicate that DNA is present. It is expressed or not, you can find only in EST, you can find in microarray, you can find in mRNA. mRNA is expression related. Got it? Clear? Yes. Hence D is the correct answer for this question. Alright. So now shall we make a move towards the next question? Everybody in the class is getting clarity on this. So now are you confident that you can really take up this exam in a light manner? Yes? Yes, absolutely. Come on everybody. Yes. Beta-lactam ring. Beta-lactam ring. It is present in beta-lactam antibiotics. Who are they? Chloramphenicol, erythromycin, penicillin, tetracycline. Very good, Nikki. Yes, Anjali, Archana. Too good. Keep it up. Beta-lactam ring. Yes, Chiranjeevi. Beta-lactam ring. Where it is present? It is present in penicillin. Do you remember which enzyme will able to break this out? It is going to be beta-lactamase. Right? So this beta-lactamase will only break them. Beta-lactam ring. Alright? Yes? Okay, fine. So coming to the next question. Very good, Ash. So good. So which one of the following amino acids it is going to have a very strong tendency? Tendency of what? It will have such a brilliant tendency. Yes, Nida. Yes, Ansh. Yes, Ram Prabha, Anjali, Archana. Very good, everyone. To disrupt the alpha helix. So it is the helix breaker. Who is that helix breaker? Is it? Yes, Vikash Mitra. Good. It is none other than proline. Apart from proline, all others they are not creating that much disturbance. Yes, this proline is a very notorious fellow. He is only going to make your alpha helix to get disturbed. He is only going to disturb. And apart from this, I'll tell you, this glycine, proline, they are helix breakers. Also, whenever there is a continuous stretch of positive like lysine, whenever there is a continuous stretch of negative like glutamic acid, they also will disrupt. Why? Because due to the presence of continuous chain, they will have the repulsive forces. Okay. So if at all it is having a continuous long chain, they will have a repulsive force. Yes. But if they are present alone, yes, Mamta, definitely. Yes, in plus I'll ping you the link, Mamta. So I have taken very important classes for enzyme kinetics. I'll ping you the link. Yes, that one you can follow. Okay. Yes, we have covered a lot for enzyme kinetics in plus. Alright? Yes, fantastic. So coming to the next question. Which of the following? It is, yes, Mamta, proline is very rigid. Which of the following is not at all the characteristic of what? Is not at all a characteristic of your collagen fiber. Collagen is a very important fellow, isn't it? So which of the following is not at all a characteristic of the collagen? Yes, Nida, Archana, Nikki, Anjali. Very good. So the first option says that, yes, Ash, each of the strand is independently stable due to the presence of hydrogen bond. Yes, Anthony, seeing it a long time. Come on. It is a fibrous protein with alpha helical, yes, Mamta, alpha helical coil in a coiled structure. It is the main component of the skin, bone, tendons and teeth. The core of the triple stranded helix is populated with glycine residues, right? So which is not a character? Absolutely, your option 1, which states that it is independently stable due to hydrogen bond, is irrelevant, and that is going to be the wrong answer and right answer for our question. Okay, so you are going to match the list 1 with the list 2. Alright, so coming to the list 1, it says that about the neutrophil, then platelets, B cells, T cells. Okay, so left hand side, what are the list they have given? Neutrophils, platelets, B cells, T cells. Right hand side, they have given about they make the antibodies. By this only it could have been done. Yeah. Okay. Then they phagocytize. Yes, Anthony, Archana. Come on. They phagocytize. So they will be phagocytizing. Yes. And destroy the bacteria. Next, they kill the virus infected cells. They initiate blood clotting. Yes. Okay, fantastic. Yes, Anthony, Siranjeevi, Ramprabha, Nikki, Anjali, Archana, Mamita. Okay, fantastic. Yes, everyone, option 1 is obviously correct because you all know that neutrophils, they are associated, yes, associated with phagocyte and they will be destroying the bacteria. And what about the platelets? Yes, Vishaka. The platelets, what do they do? Platelets, it is initiating the blood clotting. Yes, and all these clotting factors, thrombin and all, it is there, no, it is all helping out. And what about the B cells? B cells only make the antibody, they are the plasma cells. And the T cells, they kill the virus infected cells. Yes, so that is what is creating the cytokine storm even in COVID. We have seen that in detail. Okay, yes. So the next one. So there is going to be a breeding I got my pen is not working, guys, so I couldn't write for you. Okay, right? Okay, I'm just trying to write, but it is not coming off. Alright, anyhow, we can try to solve this. The breeding of AA, it is a heterozygote, and AA, a dominant one. Yes, it is going to result in 5 offspring. Okay, so what is the probability that, it is a probability, 4 offspring will have the AA genotype. Yes, so this is absolutely a probability based question. Yeah, come on everyone. So give a try for this question. Maybe you will get it right. Yes, come on everybody. There are 5 offsprings. Okay, so they are asking what is the probability that 4 of them will have this AA. Okay, right. So that is the question. Yes, Nikki. Yes, Priya. Different answers are there. Correct. Yes, Siranjeevi. The correct answer is 1 out of 32, isn't it? Yeah, because they asked that, yes, they asked that 4 offspring should have the AA phenotype. Out of 5, all the 4 should have, right? So only the probability it is going to be 1 out of 32. Sorry guys, my pen is not working, so I cannot put the solution for you right away. Yes, maybe in the next class I will try to put it for you. Right. Yes, Monica, correct. Ram Prabha, Siranjeevi, and yes, everybody, correct. So which nuclease of bacteria it is going to be responsible for degrading the genome of invading lambda phage? So in bacteria, I have some defense mechanism. Because of this defense mechanism, I am chopping and chopping all the viral entry. So what is that enzyme? Exonuclease, phagodegradase, restriction endonuclease, topoisomerase. Yes, Anjali, Ram, Sayantani, Nikki, Ash, correct. Very good. It is none other than our restriction endonuclease, isn't it? So restriction endonuclease only, it keeps it prevented from invasion of the foreign things. See the next question. Such a simple questions have been asked in DBT 2021. So similar only will be coming for your exam also. So the only thing is you have to develop fast reading. One question not greater than 20 seconds. Yes. DNA fingerprinting for forensic is based on direct, exon, intron, promoter, repetitive. Yes, everybody, fantastic. It is variable number of tandem repeat. It is repetitive. Very good. Yes, Anjali, Ash, Priya, Sayantani, Ram and Ash. Yes, absolutely. That is going to be the correct answer. Got it? Shall we make a move towards the next question, everyone? Yes or no? You are all ready for it, right? Yes? Okay, fantastic everyone. Yes, please do note it down. Yes, Monica. Good. Yes. Which of the following growth curve or growth curve of bacteria it is more sensitive to ampicillin? So in your growth curve, yes, Sangeeta. So as far as this growth curve is considered, you will have different phases, no? Lag phase, acclimatization phase, log phase, maximum growth, stationary phase, decline phase. Yes. So which phase is more sensitive to that of the ampicillin? Ampicillin is an antibiotic. Mixed response are there. I'll tell you the correct answer is only the log phase. Okay. The first option you can immediately eliminate because already it is declining. Okay. So it is not more sensitive at all. Lag phase, cell will not actually multiply. Okay. So antibiotic will inhibit the multiplication. In lag phase, cells actually they do not multiply. Which phase the cells will multiply actively? Log phase. That is why in log phase the bacteria are more sensitive towards the ampicillin. Got it? Now you understood the importance of these drugs. Yes, antibiotics. Yeah, they will be killing them or they will be showing a very big effect only in the log phase. Yes, Priya, very good. Got it, everyone? Yes, Anthony. Good. Right. So the next question is quite common question. In this ETC, electron transport chain, which one, yes Ram, which one of the following can be a 2 electron carrier? It is going to be a 2 electron carrier. Cuproproteins, cytochrome, flavin, iron sulfur proteins. Very good, Sangeeta, Sayantani, Anjali. 2 electron carrier, none other than flavin. Yes, yes, correct. Yes, Manju, absolutely correct. Flavin is the correct answer. Okay, so coming to the next question. Yes, so till now you are all happy. Yes, I think part A of this 2021 paper almost we got it covered. Yes, so one more session I will be putting this weekend to cover the part B questions. So once 2021 is fully done, you will get an idea. Yes? Okay. So which one of the following can be effectively transmitted from patient to patient or person to person through coughing? Okay. AIDS and tuberculosis, dengue and COVID, malaria and typhoid, swine flu and adenovirus. Yes, think. Yes, Sangeeta, Manju, Ash, Priya, Aarti, mixed response are there. Through coughing, which is going to be spreaded. Very good. Yes, Aarti, Monica, Priya and you are right. The swine flu and adenovirus. Yes. So if you take this AIDS, it is sexually transmitted. Dengue, it is vector based. Malaria is vector based. Cough based. Swine flu and adenovirus, they are only cough based. Yes? Okay, fantastic. Right. Yeah. So coming to the next question. Yes, everyone. So coming to the next question. I think this is done, no? Beta lactam ring is present in penicillin. We did it. Okay, I think this also we are done. Okay, we'll go to this question. Yes. Which of the following molecules they will be causing? Yes, Anthony. Yes, Anthony. They are causing catabolite repression in this lag operon. This concept, how many times you could have studied? Yes, such an important concept. Which of the following it causes the catabolite repression in this lag operon? Arabinose, galactose, glucose, lactose. Fantastic. Yes, Anjali, Ash, Ramprabha, Sangeeta, Sayantani. Yes, Monica, Priya, Aarti. Very good, everybody. All are fantastically performing well. Glucose is the catabolite represent. Already if glucose is present, why the hell the bacteria will struggle hard to break down the lactose? Yes, Vishaka. From lactose only you are going to break and form glucose and galactose. Isn't it? Yes, Manju. So you don't need that headache. No, already ready-made food is there. Why the hell you will start to prepare it? But if already food is ready-madely available. Got it? Yes? Okay, fantastic. So I think this one we did know. Yeah, I think questions are repeated. Okay. Yes, this also is repeated, I guess. Alright, next one we'll do, don't worry. Yes, next question is, during replication, DNA polymerases, what they can do? DNA replication, these DNA polymerases, what they will do? They will initiate DNA synthesis de novo. They cannot initiate the DNA synthesis de novo. They will synthesize the DNA in 3' to 5' direction. It unwinds the DNA in 5' to 3' direction. Yes, everyone. Yes, Anjali, Ram, Ash, Priya, Monica Tinka, Santani, Aarti, Ram Prabha. Very good. It cannot synthesize de novo. Why? What is needed? It needs the 3' OH. Yes, Vishaka Mitra. Yes, Nikki. It requires a 3' OH group. Yes, Ash. You need a primer. That only it will extend. But like your RNA polymerase, your DNA polymerase cannot synthesize nucleic acids de novo. It requires somebody. Got it? Yes, everyone. So shall we make a move towards the next question? Alright, come on guys, let's put hands for the next question. During translation, which of the following amino acyl tRNA binds to the P site of the bacterial ribosome? Okay, this is bacteria. F-methionine tRNA, F-methionine. F-methionine tRNA is only methionine. Methionine tRNA, F-methionine. Methionine tRNA, methionine. It is in the bacteria. So, the first the amino acyl tRNA, it will bind to the P site of the ribosomes. Which one could be the answer? Fantastic. Yes, Anthony, Anjali, Aarti, Priya, Nataraj, Nikki. Very good, everyone. Correct. Absolutely. The correct answer is option 1 or A, formal methionine. So, for tRNA also it becomes formal methionine. Complementary is required, right? Yes. Very good, everyone. Fantastic. So, shall we make a move towards the next question? Yes? Yeah. I don't feel like these questions are tough. But that moment your bulb should glow. Yes, that is what is important. So relax and prepare. Everybody can crack it. So DNA replication requires DNA topoisomerases to remove the supercoiling of DNA that accumulates at the end of the growing fork. You wish to perform the PCR amplification of a gene that has been provided in a 6 kb plasmid vector. Okay, right. Why you are not adding your topoisomerases? Why? Have you ever heard that in a PCR you added topoisomerases? No. You will not add topoisomerases to a PCR. Huh? Why, Soma? Why no topoisomerases are added? The denaturation step, it precludes means prevents the formation of supercoil. That is why you are not using the topoisomerase to remove it. The reaction buffer, it has a pH that is going to denature the DNA. So it is going to avoid the supercoil. Taq polymerase, it has to initiate the DNA polymerase activity. Exonuclease activity of Taq, it does not allow supercoil. Which one will be the apt answer for this question? Yes, Aarti. Good. Ram Prabha. Good. That's it. Others? Yes, Anthony. Very good. Manju. Ram. The correct answer is 1. They denature. No, they do not require it. This question is a direct repeat from your 2020 question paper. LI spot assay measures what? Cytokine concentration in the culture supernatant, expression of the cytokinin gene, intracellular cytokine extraction, number of cytokine releasing cells at single level. Very good, everybody. Yes, Anjali. Yes, Ash. Yes. None other than D. This isn't it? Yeah, fantastic, fantastic. Okay, next is a light of, a kind of light question. Which of the immunoglobulins they are going to cause type 1 hypersensitivity reaction? IgA, IgE, G and M. This is going to be IgE. IgE is only hypersensitive. Okay. Alright. So shall we all make a move towards the next question? Is everybody ready for it? Yes or no? Come on. So in the following table 1 and 2 it has different nucleic acids. Yes Manju. And the list 2 contains the modification. I'm going to match it. DNA, tRNA, mRNA, done. Done or what? Which is correct answer? C is correct. Yes, Aarti has given the right answer. Monica Ramprabha. C. DNA 5-methyl cytosine. CpG island, no, that is 5-methyl cytosine. tRNA pseudouridine. mRNA is 7-methyl guanosine. Capping. Okay. So now it is understandable. Now it is clear. Yes. All right. Yes, everybody. So we have come up to the tail end of the class. Yes, I hope that you all had a fruitful session today. Yes, as I told you, the prices are going to hike. So before the prices are gone up, just grab your subscription. You can make use of my code Sanarnya Live to participate in this Sanarnya Live, SARANYA LIVE. So this is the code. You have to definitely make use of the code and make an complete practice for your DBT preparation. Yes, everybody. Okay, thanks a lot, everyone. Take care and all the very